Hello, this is Dr. Srijit Karvakar, a National Postdoctoral Fellow at the Department of Bioscience and Biotechnology at Indian Institute of Technology, Kharagpur, and this is a short video of my teaching demonstration on self-assembly, hydrogels, and applications. I will briefly discuss how molecules organize themselves, what we call self-assembly, and how this leads to hydrogels and their application in biology and engineering. Let me begin with a simple question. If we just mix molecules in water, do they randomly float forever? Not always. Because of electrostatic interactions, hydrogen bonding, pi-pi stacking, and dipole interactions, molecules can organize into ordered structures. For example, micelles, vesicles, coacervates, nanoparticles, nanogels, and finally hydrogels. So self-assembly is essentially nature's way of building complex structures from simple units without external machinery. Self-assembly is hierarchical. It starts with a primary sequence like amino acids in a peptide that folds into secondary structures such as alpha helices or beta sheets. Those structures then organize into fibers or nanorods, and when these fibers entangle, they form a three-dimensional network which gives us a gel. So a gel is not just thick water, it is a structured network trapping water inside. Here we show how hydrogels form and degrade. For example, alginate forms gels through calcium ion cross-linking, a simple ion exchange mechanism. Similarly, disulfide bonds can create reversible crosslinks. Let me ask you, if you increase cross-linking density, what happens to the gel? Yes, it becomes mechanically stronger. So gel properties are directly linked to molecular interactions. So why are we interested in hydrogels? Because they mimic biological tissues. They are soft, water-rich, and biocompatible, and their applications include drug delivery, tissue engineering, wound healing, 3D cell culture, to name a few. Think of them as artificial extracellular matrices. Now, how do we measure whether a material behaves like a solid or liquid? What shows the formation of hydrogels? The answer is we use rheology. G prime, or the storage modulus, tells us how much energy is stored. G double prime, the loss modulus, tells us how much energy is dissipated. If the G prime, or the storage modulus, is greater than the loss modulus, the material behaves more like a solid. Conversely, if the loss modulus dominates, it behaves more like a liquid. So rheology helps us quantify what we intuitively feel when we touch a gel. Here, I will briefly discuss a paper by Sumit Chaudhary and colleagues where short tetrapeptides were designed to form smart hydrogels. The peptide sequence follows a simple logic: XXKC. The two X residues are hydrophobic residues which introduce hydrophobicity in the system, and the C residues, which are the cysteine residues, enables disulfide bond formation. So the self-assembly is driven by two key forces: hydrophobic interactions and disulfide cross-linking. In this study, two peptides were compared. VVKC, V stands for valine, and FFKC, F stands for phenylalanine. Valine is hydrophobic, but phenylalanine is more hydrophobic and also engage in pi-pi stacking. This difference turns out to be very important. Here we see the nanoscale morphology. Notice that FFKC, which contains phenylalanine residues, forms longer and thicker fibers compared to VVKC. Why? Because aromatic phenylalanine promotes stronger stacking interactions in addition to hydrophobic forces. So even a small molecular change, replacing valine with phenylalanine, dramatically alters fiber architecture. And structurally denser, thicker fibers usually translate into better mechanical strength. Let's see if the rheology confirms that. Figure A shows the frequency sweep. In a frequency sweep, we vary oscillation frequency while keeping strain constant. This tells us how stable the gel network is across different timescales. Figure B is the amplitude sweep. We increase strain gradually. This tells us how much deformation the gel can tolerate before it yields. In both the cases, we observe that G prime, the storage modulus, is higher than the G double prime, the loss modulus. That means the material behaves like a solid gel. At a certain point, called the crossover point, G double prime overtakes G prime. Beyond this, the material becomes more liquid-like. Now notice something important. The phenylalanine peptide FFKC shows higher modulus values and yields at higher strain compared to VVKC. This confirms that morphology suggested FFKC is mechanically stronger. Now comes thixotrophy. Here we apply high strain, the gel structure breaks, and the G double prime becomes higher than G prime, meaning it behaves like a liquid. When strain is reduced, G prime recovers and becomes dominant again, meaning the gel reforms. And this happens repeatedly. So under pressure, like pushing through a syringe, it flows. When pressure is removed, the gel is reformed. That is thixotrophic behavior. This property is extremely useful for injectability and minimally invasive applications. Finally, the biological performance. Live death staining shows predominantly green fluorescence, which indicates viable cells. This means the hydrogel network is not only mechanically functional but also biologically compatible. So we now have a material that self-assembles, forms structured nanofibers, exhibits strong mechanical properties, shows thixotrophic recovery, and supports cell viability. That's the complete structure-function biology connection. To summarize, a simple tetrapeptide design XXKC uses hydrophobicity and disulfide linkages to drive self-assembly. A small molecular modification, valine versus phenylalanine, changes fiber morphology. That change alters mechanical strength, mechanical properties dictate injectability and recovery, and ultimately, all of this influences biological performance. So in hydrogel design, chemistry controls structure, structure controls mechanics, and mechanics influences biology. Thank you for your time and attention.